What is our role in nature? Because there seems to have long been this view that there's humans and then there's nature. And nature is, when things go right, nature is pristine, and that we screw it up a lot. And I think we are kind of naive when anyone who thinks that way. And I think a lot of people do. So I guess my first question, Drew, is how do we think about ourselves in relation to the natural world?
Higher natural world or the living part of the natural world?
Choose.
Let me try and start with biology. So what I've observed is humans have different broad perspectives on biology. There's the way we look at it, sort of the world of the forms, and natural living systems exist. They don't change over time. Darwin and colleagues over 100 years ago come up with a different perspective. Natural living systems pre-exist, but they have this direct descent with mutation, diversity, selection, evolution. And note those two perspectives lead to differences of opinion within our culture. The two tribes, if you will, that result from which of those perspectives you invoke in a particular moment. And then with genetic engineering showing up a generation ago and ongoing advances, there's the rise of a third perspective, which is natural living systems exist and they either are static or change via evolution, but now we can encode our intent into living systems. And that creates a third perspective, which means you need to have twice as many conversations between the tribes because there's a third tribe. And our time is one where all three perspectives coexist, sometimes usefully or not.
But isn't it also a time in which the latter perspective seems to be gaining more ground, more importance? I mean, can we seriously talk about things like keeping our hands off nature and not intervening with species?
Well, you know, I'm born in 1970. Human populations doubled, and my colleagues in biodiversity and conservation biology report to first approximation natural biodiversity is down by a factor of 2. So human population up by 2, natural biodiversity down by a factor of 2, correlation, causation, I leave that to others. But it would seem like we are inadvertently having a massive impact on the planet.
So Kevin, shouldn't we, if we're inadvertently having a massive impact, why don't we just get out in the open and have the massive impact and try to make the planet work in the way that we think we're capable of making it work these days?
Well, one answer might be humility. Just because we think we can do it better doesn't necessarily mean that we can. But more broadly, the debate, such as it is over the question of, do we really think that there is a better? Because one of those camps says, well, the way that things are naturally is the best possible way. And I sometimes question whether we have the tools to make it better, whether we have the wisdom to make it better, but I have a hard time conceiving of the way things are as being the best of all possible worlds.
Also, the way that things are naturally, like naturally in what way? Because I don't understand what the word natural means. I mean, I literally, show me something that's natural. I have students, sometimes I send them to the grocery store. I say, bring me back something that's natural, that was like in the Garden of Eden and has been unchanged. And one kid once brought uniodized salt and some spring water. But basically, there is nothing that's natural.
You could probably find some ice cores in Antarctica that still have natural organisms in them.
Yeah, I don't have this expense account to send them up there, but yeah, or down there. Okay, so if we're so humble and we are so concerned about nature, why are you engaging as a core activity right now in trying to get rid of Lyme disease by screwing with the genetics of mice? I mean, don't mice have a right to live a natural life?
Sure, all we're doing is immunizing them. They do that on their own anyway, perfectly well.
Then why don't they take care of it?
Well, problem is that they've sort of they've evolved a truce with the various pathogens. And they've done that because they and the pathogens have been hanging around, particularly on the East Coast, for a very long time. And over time, pathogens tend to evolve to inflict less harm on their hosts, which then, in turn, defend themselves less. And so they've come to an arrangement. And we are interlopers, so are our dogs and our cats. And so we all get Lyme disease and they don't.
I should back up for a second and say what Kevin is doing and what others are doing with other species, particularly mosquitoes and malaria, are using new tools of genetics One is CRISPR, this editing tool that you've all heard about, and another is gene drive, which isn't a new tool. It's just a phenomenon that allows genes to be passed down at greater than 50% likelihood that everyone gets one gene from their mom and one from their father. This is a way of getting 99% of the same gene. And it's existed in nature. It exists. It's just been hard to manipulate. With CRISPR, it is now much less hard. And since that's the case, a bunch of scientists in various areas, and Kevin was, I think, really the first to put these two things together and say, we can use the editing thing with the drive system to change the genetic constituents of various species. And one of the things that would be nice is white-footed mice are the main transmission agent of Lyme disease. I live in the East Coast. Lyme disease is something that petrifies people there. It's not malaria, but it's bad. And if you broke the chain of transmission by making white-footed mice resistant to Lyme, you'd end it, which is what Kevin is trying to do. But that's a great thing. I don't think I've been around you a lot. I don't think I've seen a lot of opposition. But maybe I don't like the opioid crisis. So why don't we just use gene drive to eliminate all poppies in the world? Why not?
Last time I checked, fentanyl was a problem, and it's fairly readily chemically synthesized, so I don't think it's actually going to solve your crisis.
You don't think there would be a benefit in getting rid of bad things?
Depends. We're talking evolution here, right? And economic systems evolve and adapt as well. Cultural evolution is also a thing. So just like when we were trying to target a virus such as HIV, you need to think about what are its escape routes, and can you close them off? Because if you just throw something at it and it's going to evolve resistance almost instantly, why are you bothering?
True, but as with targeted therapy, there are ways to surround the HIV virus. There are ways to surround lots of things. Is there a case to be made for let's just look at a species, decide we don't like it, and get rid of it?
Well, one of my go-to examples in questioning whether the natural world is, shall we say, the height of all goodness, is the New World screwworm. So this is a species that it is literally a flesh-devouring maggot. Its life cycle consists of, it's a fly, it lays eggs near open wounds. The eggs hatch, the maggots crawl in, and unlike maggots that have actually been used historically to cleanse wounds, those are from species that don't eat healthy tissue. This one does. And it's so excruciatingly painful that for a physician to simply conduct an examination on someone who is screwworm infected, they've had to use morphine. So this is a species that exists, its very life cycle requires it to cause horrific agony to higher mammals.
And you know for certain if we got rid of screwworm, there would be no unintended consequences that would be a problem.
Well, we don't know anything like that for certain, but we have already gotten rid of it throughout all of North America. And we did so by invoking that other bugaboo. We took lots of screwworm flies, deliberately raised them, and irradiated them. So this destroys the chromosomes in their gametes for the next generation, so they're sterilized. And then you release the sterile screwworm flies in the environment. They mate with the wild ones and have no offspring. It's called sterile insect technique. Started way back in the 50s, and so there hasn't been screwworm in the United States since, I think the last episode was the 70s, although they recently reinvaded Florida and helped drive the deer on the Florida Keys that much closer to extinction lately. But the upshot is they're only present in South America. And while the sterile insect technique is probably not powerful enough to do the job in South America, just because the geography is much more challenging, with something like a gene drive system that could spread infertility through the screwworm, that could probably do the job. And if you want to know any given environment, is it a good idea? Well, the US Department of Agriculture funds a facility in Panama that produces something like 5 million sterile screwworm flies every month.
So if the US Department of Agriculture does it, it has to be a good idea.
Well, you know.
No, but interesting.
If you want to know in some ecosystem, does it cause a problem, then drop a couple million sterile screwworm flies there. It'll be locally eradicated. Watch the ecosystem. Does anything bad happen? If not, you're probably okay.
So sterile insect technology, another way of looking at this is to use some much hated words, genetic modification. And a few years ago, I went to a town meeting in Key West, Florida, where a company then called Oxitec was presenting in data. They wanted to introduce genetically modified mosquitoes to get rid of dengue fever into Key West because dengue had gone away and it had started to come back. It's a bad thing. I had been to Brazil with these people, and the Brazilians were delighted. In Key West, they were treated like murderers. I mean, it was ugly. Like, you're trying to invade our town with this genetically modified stuff. What they do to get rid of their mosquitoes is dump chemicals in their backyard. Somehow that was okay. But my question really is, and Drew, I know you've thought about this a lot, where is there a line? What is it that makes people uncomfortable about using chemicals to make new things, changing the way nature is in the laboratory? Why is it seen as unacceptable to do that?
It came up a little bit yesterday in the conversation between Walter and Jennifer, and so far as I can tell, and there's probably many reasons, but one that's most on my mind is the discomfort associated with taking responsibility for transmitting your intention into some object. And it's one thing if you're encountering biology and you're breeding an animal, or you have some kids and you're using genetic information to decide from a sequencing experiment whether or not to bring a child to term, you're dealing with the cards that you've been dealt by nature. And so it may be good or bad, but it's not really your doing. You're just responding to the things that have been presented to you that we inherit. But as soon as you go in there and you say, I would like that nucleotide to have that letter, A, T, C, or G, and I'm transmitting my intention into the genetic material, you simply can't hide from the primal responsibility of a transmission or encoding of your intention into the object, and that then leads to all sorts of very normal cultural responsibilities that we're familiar with. And I think people are very nervous about making that transition for a number of reasons, but that's my sense of it.
What can we do to make people a little more, is there anyone out there who's uncomfortable with the idea of people using CRISPR in laboratories and on various species? About half. Oh, I'm sorry, CRISPR is a bacteria. It's been around forever. It's a bacterial defense system. And it was discovered by mistake in 1987 by some Japanese scientists who were looking at the sequence of E. coli, a bacteria. And they saw this weird set of nucleotides that were a palindrome, the same backwards and forwards. And spaced in between nucleotides that they could recognize were these weird things that they did not recognize. And I won't go through the whole thing, but eventually people realized those weird spacers were broken up bits of viruses that had tried to attack the bacteria. And this was a defense system. The bacteria basically gets the virus, chops them into bits, incorporates some of it into their immune system so that the next time they come back, they're ready to fight. It's kind of what our immune system does, but nobody ever thought bacteria had a sophisticated immune system. So that's what it is, and a bunch of people, most notably Jennifer Doudna, at a certain point said, if this bacteria is so sophisticated that it can attack viruses, we can use it with special pieces of RNA to go to places in the genome where we want it to go. And people have been able to basically turn CRISPR, a defense system, into a GPS in your genome. You can decide what group of nucleotides you want it to go to, and it will go there, and it can replace things. And so it's a kind of cut and paste system, somewhat simplistic, but it's kind of like a Word document on your laptop. You can change words around, you can change sequence around, and when you start thinking about doing that, there's a lot of very exciting and a few horrifying things that can be done. That's CRISPR.
Can I give a compliment to that? So it's a fourth generation genetic engineering technology for cut and paste. It's programmable in a very easy way that makes it much easier to use. And I was interested to see the hands that came up regarding use of CRISPR in laboratories. If you go to a website, The Odin, you can buy a CRISPR kit for do-it-yourself CRISPR, do-it-yourself genetic engineering. So one of the interesting puzzles 40 years after the birth of genetic engineering with the early tools is we have a second demographically, we have a second generation of genetic engineers arriving. And the tool platforms are just much more capable and shareable.
Yeah, this isn't the first of these tools. But Hank Greeley, who's at this meeting, maybe is in this room. Hi, Hank. Once said to me, CRISPR isn't, it's like the Model T. The Model T wasn't the first car. It was the first car everyone could have in their driveway and use, and it cost a reasonable amount. And CRISPR's just a much easier system to use than the things that came before it, and it's therefore being used in every lab.
So what is the answer to this example?
To what example?
Should we field deploy a genetic drive to get rid of this?
Well, let me ask, and I love participation. Who thinks it's a bad idea to do the type of thing I just described Kevin's trying to do to get rid of Lyme disease? Is there someone who thinks that's dangerous or bad? Is it specific to what?
So what we're proposing is to take antibodies. So the mouse's immune system generates antibodies that will specifically recognize either the tick saliva or the Lyme pathogen itself. We are taking those antibodies from the mice, we're identifying the genes that encode them, and then we're putting them back into the mouse genome so that, unlike how it normally occurs, future generations of mice will inherit them. So we're working with the citizens of Nantucket and Martha's Vineyard on this. They're in charge of the project. They have steering committees. They told us, yes, we want both anti-Lyme and anti-tick. Do both, but we don't want any, they said we don't want any foreign DNA at this stage. We just want to know if it works using just the native mouse antibodies. So the resulting mice will be 100% mouse. Give us a bit of context. How many of you ate beef in the last week? Please raise your hand. Did you know that the genome of the cow is roughly 25% snake? Because a couple million years ago,
I really can't take you anywhere.
a virus moved a happy little bit of DNA that is one of those natural forms of gene drive that Michael referred to earlier, from a snake into the auroch, the ancestor of cows, where it, being a gene drive, replicated itself within the genome until it's about a quarter of the cow genome. So the point is that what we call species, that's an arbitrary human conceit. Our minds like putting things in boxes. We say this is a species, that's the other, this is a different thing. Biologists cannot agree on the definition of a species. Different subsets of biologists have different definitions. And nature violates these boundaries all the time. But that doesn't matter, because the citizens of Nantucket and Martha's Vineyard live where they live. It is their environment, and they are in charge. So they said, okay, we get that, but we're still going to feel more comfortable if there's no foreign DNA in these mice as we define foreign. So at which point we say, okay, that's what we're going to do. So we are building mice that are 100% mouse, express native mouse antibodies, and the plan is, if field trials on mostly uninhabited islands go well, then perhaps 7 or so years from now, these citizens of Nantucket and the Vineyard will vote to decide whether or not to release about 100,000 genetically engineered mice onto their islands. And if that happens, then the mice will transmit that immunity to the entire native mouse population, because mind, these are islands. There's only so many mice. There's not new mice invading all the time. And those mice then will kill ticks that bite them, so there will be many fewer ticks. And any ticks that they don't kill, they will cure of the Lyme pathogen, because the mouse immune factors will attack it even within the tick. So it'll disrupt the cycle of transmission, and the rates of tick-borne disease will ideally plummet. We can't say exactly how far, that's what the trials are for, but that's the idea.
We're going to take questions a little later, just because I'll lose my mind if I start answering, if we do it. We'll do it, I'll give a bunch of time. I'm sure there'll be a bunch of questions. I'll bet what you were kind of asking in asking that question is, are there some sort of possible consequences or impact that we don't necessarily know? And I think that's a good question. It's an easier question with the work Kevin's doing on Nantucket, because he's built a system that's great, because they're using mouse DNA and it's an island. So you are just looking at the mouse. But there are scientists who are very far along in editing the mosquito Anopheles that carries malaria. And they want to edit that mosquito and release it in Africa so that it will, in perpetuity, mate, and every time they do, those subsequent generations would be unable to transmit malaria. Now, there are some questions to be answered that are unanswered, and possibly unanswerable. But I will tell you one fact. 750,000 kids die of malaria every year. 200 million people get sick, have very disrupted lives because of malaria every year. It's one of the greatest causes of sickness, loss of economic earning power in the developing world there has ever been. So that's got to be considered when you also should think, what are the unintended consequences going to be? There may be some, and they may even be worse than malaria. But you'd have to have a better imagination than I have to understand what that worse would be. So I kind of really feel that when we're talking about what we're talking about, dicking around with evolution, changing the genetics of species, you have to ask yourself not just, is there going to be an impact? What don't we know? But also, what do we know? What do we know will happen if we don't do it? Because I think that's an unanswered question most of the time. And I think the answers to that will be different, depending upon what you're doing. There's a lot of talk about using these sort of systems to get rid of Zika. They have 2 really good Zika vaccines that look great right now so far. And I would argue, are you going to change the genetics of a species rather than use a vaccine if it works? But these guys might think that's not.
I think it's also a broader topic, right? So I mean, public health, vector control is a valid area of interest and activity. But to return to the earlier framing you offered, nature, what's our relationship with nature, evolution, and so on. Invasive species, preservation of natural biodiversity, islandconservation.org, as a charity that seeks to eradicate mice and other creatures that have taken over islands and deplete the reptiles and birds. They use poisons. They carpet bomb islands with poisons, and sometimes it works and sometimes it doesn't work. They'd like to, have a gene drive that's much more active in a mammal and zeroes out a mouse population, perhaps. One of the things that Leo Szilard, the physicist, he wrote 10 commandments, and one of his commandments was, don't destroy what you can't create. And if you take a slightly deeper view on some of this, I think a lot of these questions you're raising don't go away ever, but they become mitigated in part because you can anticipate being able to create species or construct them at least. We've seen over the last decade several different projects to synthesize entire genomes, not edit genomes, but synthesize from information and raw material intact genomes and then reinstantiate lineages. All of those thus far have been re-implementations of natural examples. But if you look at how the research community is developing now, it seems like we're about 5 years out from non-trivial examples of reproducing microbes that are not descended from natural examples, but are just new. The Dutch, for example, just got organized and invested 200 million euros in a build-a-cell project. It won't be a creature, that you see be a little microbe, but microbes are the first cells, and then you make other things from that. So it's not hard to imagine in the near future we're actually reasonably good at building creatures, and then what, right? And you go back to island conservation. There's sort of an example of one of the more interesting connections between the world of biotechnology and the world of nature, where the conservation community, struggling to figure out how to maintain natural biodiversity, is slowly recognizing some of the leadership, very controversial within this community, that you can't get back to 1850 and what nature looked like then. You can't get back to 1950. You got to look into the future and say, what 2050 do we want to get to? And what does that look like? And what relationship do we want to have with the tools that Kevin or others in biotechnology have? And I don't think anybody's figured out that conversation yet, but it's an amazing connection being developed.
A lot of people don't want to have the conversation. Kevin and I went to the annual or quadrennial IUCN International Union for Conservation of Nature meeting in Hawaii last year. Hawaii's birds are disappearing. By 2050, all the native species will be gone. The Audubon Society calls Hawaii the extinction capital of the world. There are ways one can envision, and the main reason is that there's a form of malaria called avian malaria. It's killing the birds, and it's spreading higher and higher because despite the Trump administration, there seems to be changes in the weather that allows these insects to live at higher elevations. So How do you get rid of those insects? You can't really spray them, which is what we would normally do. They're in rainforests. There are many protected species that would also suffer. You could use some of the techniques these guys have been talking about, and it ought to work. There was tremendous debate in the conservation community, which is still going on, and many people just say, well, it would be better to let all the native bird species of Hawaii die than to intervene in this way, to change the genetics in this way. I have to admit, I do not understand that, but there are many people who feel that way.
Well, it's a particularly interesting one because we brought mosquitoes to Hawaii. That is, if you went to Hawaii a few hundred years ago, there would be no mosquitoes at all. So we introduced them, and a few decades back, avian malaria came along too. This goes back to this question of, is nature good? Well, it's not just us that have a malaria of our own. Many different species have a malaria of their own because there are lots of parasites and pathogens in nature that evolve. And evolution doesn't care about that. If it replicates, then it will proliferate and it will be there. That's what evolution does. But the mosquitoes were our mess. We did it. And the question then is, should we use our technology to remove the mosquitoes? And who gets to decide that is a very important question. Is this something that should be up to the native Hawaiians? Is this something that should be up to all the citizens of Hawaii? Should people who don't live in Hawaii but just visit there get any say in this? And
why isn't there a massive lawsuit against somebody regarding something to do with the introduction of the mosquitoes, or the Navy having to do with something associated with the introduction of avian malaria?
Don't give people ideas. Probably because it happened long ago enough for there not to be.
Everyone's dead. I don't think the heirs of those corporations have any assets left to seize, unfortunately. Also, it's not really going to solve the problem. But in this case, we could conceivably remove those mosquitoes. Now, you've got to be cautious.
It may establish agency, right? Which is the point of your comment.
Well, but this is interesting because it would establish agency. But in this case, this is a mess that we, or at least humanity, made. And now the question is, are we obligated to clean it up? Another interesting question is, are you obligated to clean up a mess that you didn't make? So there's this famous example of what do you do if you're walking along, you're wearing some very nice clothes, and you see a child drowning in a lake? And you know how to swim, so do you jump in and save the child and ruin your nice clothes? So Peter Singer put forth this argument a long time ago. He said, well, of course you would. Now, you didn't throw the child in the lake, but you're still obligated to jump in and save them. And so he said, well, then, if you're willing to do that, why aren't you willing to donate the cost of your clothes to save a life on the other side of the world? Because there are children dying there. This is a great way of making all of us feel like very bad people.
He actually went further. He said, and if two people are in the lake drowning and your daughter is over there dying of something, you are obligated to save the people in the lake because there's two of them. And your job on Earth is to increase happiness as an aggregate. I like Peter, but disagree with him on a couple things. But it's a very complicated question of when you intervene.
But here's the thing. If you don't know how to swim, the first rule is you don't endanger your own life trying to save others who are at risk, because you'll probably only get yourself killed and fail to do the situation any good. So if you don't know how to swim, you're off the hook in this dilemma. You have no obligation to do something. But as soon as you learn how to swim, then it becomes a test of your moral character. These technologies are allowing us to swim. So we didn't create, for example, suffering in nature. We brought the mosquitoes to Hawaii, but we didn't make the New World screwworm, for example. Evolution did. But now that we can do something about it, we may choose to intervene or we may not. But morally, it's on us because we have learned how to swim.
Depends on our morality.
Well, speaking of morality, so does anyone know what God's 8th biblical plague was? What? Locusts. Plague of locusts. So my friend Kevin recently said, I would like to have on my tombstone, I tamed God's 8th biblical plague. Which I think I'd love to put that on your tombstone. Really, no, not tomorrow, but soon.
Thanks, Michael.
But the question is, we didn't That's not our fault that there's the locust. Why do you want to get rid of that? I mean, where does it end?
It's a great question. But in that case, if farmers that live in some of the most deprived and impoverished regions of the world would prefer that they not suffer 100% crop loss and total devastation when the plague of locusts screams down and eats everything in sight, as they have been doing since the beginning of recorded history, can we fix that situation without harming the locusts? In this case, potentially yes, because the locusts are most of the time just desert grasshoppers. And then it rains in the desert blooms, the locusts eat all of it, they reproduce rapidly into massive numbers, and once it's all gone, that proximity causes them to switch from being solitary, largely avoiding each other, into gregarious. So then they actively form a swarm. Their dietary habits change, their breeding habits change, and the swarm roars out of the desert and eats everything in sight. That is a genetically programmed change. That is, that switch from being a solitary grasshopper to being a swarm. Using a gene drive system, we could disrupt that. That is, we could switch off their ability to swarm. And we know from studying locust genetics that there are perfectly stable populations in the desert that maintain in solitary mode indefinitely. So the species would still be there. They just wouldn't cause problems outside the desert. And one of the lovely things about editing DNA is that in this case, if we decided we wanted locust swarms back at some point, once we no longer were vulnerable in terms of impoverished farmers who needed their crops to survive, then at some point we could turn it back on. So should we do that? That's a complex question. And I think, personally, I would say that's not for me to decide. That's for the people who live in the region to decide. But it's also a very complicated one because these locusts affect roughly 20-some countries. How are those countries going to agree? How are the African countries affected by Anopheles going to agree? So,
big difficult question. I have another weird theoretical question. It has to do with how far down into the future we should look. So sickle cell developed as a way of keeping malaria out of the bloodstream. And I think 200,000 or so years ago, if we had all this cool technology, everyone would be sitting up here saying, we've got to engineer sickle cell into everybody so that nobody would get malaria. And then everyone would die of sickle cell disease. It's a slightly simplistic view, but the point is, it's really very hard to look endlessly down the field and see what's going to happen. Now, I think Drew would probably have a response to this.
Well, no, in The Little Prince, the fox is teaching the boy, and there's a back and forth that basically gets to men have forgotten this truth, humans have forgotten this truth, but you must never forget it. You become responsible forever for that which you have tamed, right? And Kevin's example of the locust is a fine on-ramp into that world. If we're going to intervene in these ways and assume a type of morality and a type of responsibility for our actions based on intention, then we have an ongoing responsibility. Final exam in the Introduction to Bioengineering course at Stanford a couple years ago had this as the first question. And we talked a lot about editing DNA, but I think we should talk a little bit about synthesis of DNA. So there's a technology called DNA synthesis, and you can think of it like a musical synthesizer, but instead of having 88 keys on a piano keyboard, it just has 4 keys, A, T, C, and G. And depending on how you press those keys, a machine will dispense chemicals that makes from scratch a piece of DNA. So you're just printing DNA. You're not editing it. You just make whatever piece you want. And this is a technology that was established in Colorado circa 1980, but has been getting better and better and better over time. So when I first started teaching in 2003, it cost $4 to press a key once. But I just signed a contract this month where it cost me 2 cents to push the key. So the cost down between 2003 and 2017 is from $4 a letter to 2 cents a letter. So the final exam question at Stanford is the following. The cost of tuition at Stanford is approximately $50,000 a year. It's got this unfortunate trend of increasing over time. It's doubling about every 15 years historically. But the cost of printing DNA is dropping by about a factor of 2 every other year. And that trend, and then there's this thing called the human genome. And to make the math simple, let's presume the human genome is about 4 billion bases or letters. And so the question we ask our engineering students is, we've got this increasing cost of going to Stanford and this decreasing cost of printing DNA. At what point in the future will the cost of printing the DNA and coding an entire human equal the cost of a year's tuition at a private university? And it's a mathematics question with the two logarithms. You just have to find the intersection, and we grade the students on the math. And the answer is 19 years from today. And I think the significance of that is less than one human generation. And that requires massive improvements in capacities and technologies, totally non-trivial. So the standard disclaimer, like past results don't guarantee future performance. But part B of that question is the following. Do you and your future partner, if you wish to have kids, start saving up money for college tuition, or start saving up money so that you can print the genome encoding your lineage, your offspring? And we don't grade that on any particular answer, just the reasoning of the answer. And that one splits the class. About 60% of the students say, this is wrong. It's not what it would mean to be a parent to print our offspring's DNA. And education is special, so we're going to save up for tuition. But 40% of the class say, the cost value of education is going to change. There's going to be Khan Academy 10.0. Stanford's not that great. And meanwhile, if there's any chance this comes true, and it could give me the option of securing some advantages for my lineage, I might as well lay in preparations to secure that option. So I'm going that way. So I think there's, if you allow a little bit of time scale here, and the time scale isn't that much, we're talking of order a human generation, the possibilities are much stranger than I think we're prepared for. And the institutionalization of what's going on the basis of the politics of CRISPR and sort of assimilating all the culture into the existing institutional frameworks based on modification kind of misses the point.
Don't you think it's just an intermediate step? I mean, it's hard to go just from nature to make your own human without something in the middle. Wouldn't you just see this as maybe something in the middle?
Yeah.
Is there anybody in favor of making your own human in this audience? Really? I definitely would do it. What about you? Would? Yeah.
Not that way.
What way would you make your own human?
Probably taking genetic material from some number of people and doing what's called iterative embryo selection.
So conventional.
You know, basically do every human generation in a dish every couple of weeks or less. Sequence all of your candidates, decide which alleles you want, combine them. That way you're not taking a major risk because you're not introducing any DNA that would not have been in some human before. But you can do that now. Well, you can do it in mice probably about 1 to 2 years.
Read Hank's book. Yeah.
Yeah, we're stepping on End of Sex by Hank. Every single person in this room should read it. It's required.
Yes.
And we're stepping on his ground a little. And yes, I'll take 10%. But these are things that people need to think about, and I don't think we have thought about them. And partly because I personally don't think there's a literacy to think about these things. I would love for kids in 3rd grade to be editing genes in worms. Turn a worm from green to blue. Get excited, the way I did when I blew stuff up with my chemistry kit when I was a kid. I was bad then, and I'm bad now at that. But some percentage of people get really excited, and even those who don't and aren't good, like me, see the point of it. You can't see the point of these things if you have no real knowledge, and I don't think this knowledge is taught or approximated or communicated very well in schools. And I think that has to change before any of this.
Well, we can quantify that a little bit. The iGEM competition that we started years ago now it's like the robotics competition, but for genetics. It has 6,000 students a year. It meets every fall in the Heinz Convention Center in Boston at a big jamboree. So 300, 400 teams of students from universities and high schools all over the world. And 6,000 students might seem like a lot, but then if you think about those students having careers that last 30 years, that's 180,000 people. And maybe there's 1 million people on Earth who are actively literate with reading and writing DNA who could program or hack DNA. And so that's 1 million out of rounding up 10 billion. So that's 1 in 10,000 people. Meanwhile, over at the United Nations, when we talk about a different type of literacy with human-to-human languages, like English, we have millennial development goals of everybody should be literate. Right? So a side question, which maybe we pick up in the afternoon, is what fraction of people on Earth should be actively literate with any particular technology? Programming a computer, programming an organism, pick your favorite technology. And if you want to get to citizenship, I think the literacy rates have to be much higher than 1 in 10,000.
I agree.
But then, along with that, there's a conceit reflected on the stage, which is the decision-making process will be informed by thoughtful organizations, communities, and groups, like what Kevin is pioneering in an amazing way on Nantucket and so on. But unilateral actors, right, individuals who I think this is important, so I'm going to do it, probably win, I would say, over a longer period of time, because they just get to do it.
Depends very much on the nature of the technology. So with gene drive, for example, the problem with it is that the current simplest, easiest to do form, so CRISPR is encoded by genes, and the instructions telling it where to go in the genome can also be encoded in the genome. So a CRISPR-based gene drive simply says, instead of delivering the CRISPR components from the outside, let's do that. Let's deliver the DNA we want to insert, the CRISPR encoding that insertion that says cut here and replace with this sequence, but also encode the CRISPR system, the instructions for making that change. So you put that in the organism. If you make it heritable, then you have released a find and replace into the population. Because when that organism mates with a wild version, in the offspring, they'll inherit 1 copy of the edited version and the CRISPR system for making that change, and 1 copy of the original wild version. And the CRISPR will cut the wild version, copy over the new version. So now all of the offspring are going to inherit.
I see there's these little graffiti things on the trees out here, kind of a shame every now and then. But I'm just going to put my name in some DNA code and hop it on one of these gene drives and release that so that I've got Drew in the DNA all over the planet, right? And I presume other script kids will do that.
And right now, designing this is trivial. Middle schoolers could do it.
All right.
Now, they can't actually make it happen, because delivering the DNA into the reproductive cells of an organism that has sex, because this only works through sex, is incredibly complicated, and only a few people can do that.
I didn't.
A warning beep.
Well,
But here's my point. This is a one-to-many technology. It's a unilateral sort of thing. Someone can just do it. But it is slow. It is unfailingly detectable if you bother to sequence the DNA. And anything that you can find and replace in your Word document, if you don't like it, you can run another find and replace. So it does provide some kind of veto power to society more broadly.
It does, but I think, I've never said this before, but if you want to hear how this is resolved, come to our session in the afternoon. But I think there's a lot of questions, and I feel like we should let people out there participate in this a bit. So I'm going to throw it open. And people, there are mics, and there's a woman in the back. And we'll just...
Hi, I have a question. So it sounds really great that we can get rid of malaria and Zika and all these other viruses that are born from mosquitoes. I guess my concern is that there isn't some other super virus lurking in the background. Have we studied mosquitoes enough to know that okay, these are the only viruses that they're going to produce. And if we get rid of these, it'll be okay because we've studied them to know that they can't evolve into this other super virus that would be worse than malaria and Zika, even though malaria is a terrible thing, especially in third world countries. I mean, it probably would be hard to get a lot worse. But have we studied the mosquito enough to know that turning off this isn't going to turn on something else? I guess is my concern.
The ultimate answer is we're not turning it off. There's 3,500 species of mosquitoes. There's 5 that we're talking about that have the ability to do this. So we're not even talking about dealing with most mosquitoes. If your ultimate question is, can we finally know if doing this will be beneficial without a risk, the answer will always be no. I've never heard of an entomologist who's found any reason to believe that this would create the ability for those mosquitoes to do something worse. But it doesn't mean that couldn't happen.
It actually cuts to the heart of a question as to the appropriate approach, because one is to make the mosquitoes resistant to the pathogen, so they just can't transmit it. The other is to suppress the mosquito's fertility. The mosquito population goes down to 1% of what it was before. That's the frequency of biting someone with malaria, getting infected, and then going on to bite somebody else and infect them would then be too low. And the advantage to that approach is it doesn't matter what other viruses might be out there, you just don't have those mosquitoes around to transmit them at high enough frequencies.
But then the question becomes, is there another insect that's worse? And then you presumably go to data that says in places where we've gotten rid of mosquito, we haven't seen that niche reoccupied.
And that, in fact, is true to the best of anyone's knowledge. Maybe, I'm sorry. We're going to work our way down, I guess. Yes, we'll get you, I promise.
I'm a novice in the sense that I find all of this very interesting and confusing. Yesterday, we had several sessions on how you alter genetics in humans and the way you go into stem cells and can change a person from being even from a male to a female, and that you don't have to, you can actually go to the skin and create embryos. Can you sort of explain the difference between what you're doing, stem cell therapy, DNA, genetic engineering, and how they come together? I mean, you've basically been talking about animals, but how does all of this apply to humans as well?
Yeah, it's a good question. I mean, people are really good at thinking about people. We're selfish as a species. And so the great sessions from yesterday were really focused on humans, human reproduction, editing of humans. What we're bringing to the table is there's other living things on the planet. And in addition to what we wish for ourselves, how do these same tools relate to the other things on the planet, the other creatures and critters, and what do we want our relationship to be with them as well? And at the core, from a toolkit level, it's the same toolkit, right? All of biology to first approximations running on DNA, so anything you can do to edit or gene drive or synthesize DNA is generally applicable.
With one very important caveat, a gene drive only spreads parents to offspring.
Yeah.
So it takes generations. So anyone who tried it in humans would need to wait a couple thousand years for the results.
I think one of the things that was hidden yesterday from the morning session in particular, it was a great conversation, but Walter and Jennifer stopped in 2015 in terms of citing the state of the world, but we're obviously in 2017. And so what they related was, for example, in 2015, the US said moratorium on germline engineering of humans using these tools. And the work that had been done in China was on non-viable embryos, so things that could not be implanted and brought to term. But the news from this spring, just a few months ago, is the Chinese have now shown the techniques work in viable embryos. And the US government has issued a report saying, okay to go ahead with modifying the human germline for traits that are medically essential, if you will. And the moral reasoning of that government report of the US is approximately the Swedish are doing it and the Chinese are doing it.
Yeah. And by the way, this was like a universally agreed upon tenet in December 2015. Do not screw around with the human germline. So, 18 months later, we're all doing it.
But in one of the morning sessions, they also talked about the way the Germans tried to genetically alter the humans back in the 30s, and that Israel, to some extent, is doing this today, which is sort of a total different view. How does that all fit in?
Well, I mean, if you're talking about making a master race or any kind of race, you need to inherit that stuff. So assuming we could edit embryos and that those embryos could become people and pass on whatever traits we decide were super people traits, how many generations would it take before that mattered in any significant way in a society? It would be, I don't know, at least 1,000. Now, 1,000 mosquito generations or 10, those happen like that. But we're talking about 1,000 years of humanity, so I kind of see that particular thing as an argument that isn't worth having.
I disagree with that. I think the editing era will pass quickly. And as soon as you get into the synthesis era, you're not constrained by lineage.
Okay, so if you're talking about just like whipping up humans in the kitchen.
Can I just offer another anecdote, right? So sequencing and reading DNA is very important because if you don't know what you're working with, how do you edit or synthesize? And a personal anecdote, I've got 2 boys, 1-year-old and 4-year-old. With the 4-year-old, it was just becoming possible to take a blood draw from a pregnant woman and sequence the DNA of the fetus because the fetus sheds DNA into the blood normally. And we knew the people who had pioneered that work, and so we thought about doing it, but we didn't do it because we didn't really need the data. But with our 1-year-old, at least in the Children's Hospital in Palo Alto, taking a blood draw 10 weeks post-fertilization, so before you can get good imaging data, really, we learned that my wife has Y chromosome DNA in her blood because we're having a boy. And it had become standard of care. It was normal practice in Palo Alto to be offered the diagnostic. It costs $700, and you sequence the DNA of the fetus. So that's a 3-year window where you go from something not existing, but cost down of sequencing moves out fast enough that it now becomes standard of care. And it's very hard to understand how quickly things change when you have technologies that are moving on these double every other year sort of trajectories. And it's very easy to misinterpret where you might get, because a little bit of uncertainty amplifies into, well, it was much later or much sooner.
That's true.
But I suspect this is moving with real pacing.
I take this question with great trepidation, but Christina?
So it's not fully baked, but I have a provocation for you.
Really?
So on this question of should you synthesize your babies or send them to Stanford? I guess, so I had the opportunity to go to very fancy schools that 50 years ago I would not have been allowed to go to. I think all of us here, anyone who has the opportunity to be in this room, has been pretty successful and had a lot of really great things happen to them and have been successful, have had a leg up. Would you say that we are genetically superior to people who are not in this room and are not as successful as us? I ask that to say, what does it mean to give your child a leg up genetically versus with opportunity and just being rich and being able to send your child to Stanford?
Good question. I'm totally ducking it. Guys?
Thanks for the question. To the first part, I would say I don't feel particularly, circumstances of birth are qualitatively dominant, I would say. And just start with that. We all benefit from a diversity of backgrounds, but I think we're privileged in a lot of physical ways. I don't have a good answer to the second part of your question. I'd point you, and I'll admit to a type of massive diversity. When people start to approach that, I'll point you to Matthew Luau's work out of New York, a bioethicist. And he looks at these, I was shocked to learn this. He looks at these capacities so far as humans go, and then makes the following proposals. Let's engineer humanity to be shorter. All the conversation yesterday was like everybody's going to want to be taller. But no, his proposal is let's make everybody shorter. And oh, by the way, let's make everybody allergic to meat.
Because it would be better, there would be fewer methane, there would be less methane in the environment.
His argument is basically climate change and reduce the carbon footprint of humanity. 100 years ago, we were 15% shorter. So let's go back to that. Less atoms in our bodies, less energy, less carbon. And don't eat meat, prescriptive. So I wouldn't expect to see homogeneity. I hope not, especially around this. When I asked him how short would you go, his response was not so short that you would be eaten by your cat. and then I said, and will you go first? Will your lineage go first? And he wasn't sure.
Crazy. Oh, go ahead. Yep.
Is this on?
Yeah, probably.
Or just hit it. It's on.
Stand up and speak. I'm told to tell you.
Well, after that question, I don't mind asking my silly question. But anyway, what is the future of immunology programs fighting cancer? And are you all working on that in humans?
So personally, no. I have a couple of good friends who do. There's been a lot of excitement recently on T-cell therapy, essentially engineering your immune cells to recognize a particular biomarkers present on the tumor that is currently threatening your life. And that's very promising, of course, because the immune system is, in effect, it is the military of the body. And its job is to defend the body against threats. If you tell it to kill a particular type of cell, then it will do that. We're still in the fairly early stages in the sense that you have a tumor of your B cells, which is another arm of your immune system. All B cells have particular markers. If you program your T cells to kill anything with that marker, then they will get rid of all your B cells. And your cancer will be gone, and this works fairly remarkably well, downside being that then you have no more B cells. And unless you can remove those T cells, you can't have B cells again, because they will always be destroyed. So we're in the very early stages, but it is promising. The downside is that cancer it arises from us, from our own cells. And so targeting it in a specific manner where you target the tumor and not all of the healthy cells as well is always going to be a challenge.
But there are people making great progress. One of them is Wendell Lim at UCSF, and he's working on just this problem. How do you get rid of the bad cells without harming the cells we need? And it's early days, but there's tremendous progress. And it won't, in the end, just be cancer. It'll be chronic diseases. It'll be diabetes. It'll be many things. There ought to be a way to park cells in your bloodstream, and they should act as plumbers. And when something goes wrong, they'll fix the leak, and then they'll hang around. And when you have a leak again, they'll be there to fix it. And that's how we deal with vaccine-preventable diseases. And this is a possibility, but it's not happening tomorrow. Oh, Carl, sorry. I should point to the people with the electric power.
I have the talking stick. I just wanted to follow up on the comment you made about making people short and making them tall. Because yesterday in my session, when we were talking about people being engineered to be tall, we were sort of thinking about it not as a government deciding it would be best if everyone was short, but as consumers, parents, who would want their kids to be tall. And I think that if you look at, the demand for growth hormone in kids who are not dwarves, but are, just short, I mean, we already have that sort of consumer pressure, and it's hard to see how that wouldn't continue to drive. But the reason I bring all that up is because I'm sort of wondering if you could talk about how you see whether these things would be imposed from the bottom up by consumer demand, or from the top down by technocrats who decide this is all how we should be, or some combination, because the traits that we'll end up with, if any, would be different, tall or short, for example.
Kevin?
So I think it's going to vary by country. And one of the big questions is, if we are concerned about people who already have a leg up giving their children an even better leg up, then that's something that we should be concerned about and potentially consider making this more widely available. But I can't see our society denying people a choice. I think that's very difficult for me to imagine. And I say this with some trepidation, because if your doctor comes to you and says, we can ensure that your future children will not be on the spectrum, but they're on average likely to be somewhat less creative, I think most parents would take that, and we would lose some neurodiversity that is fundamentally enriches humanity. So consumer choice can very definitely go in some unfortunate directions.
There's another wild card. I agree with the question and Kevin's response. I just want to offer something in addition. So Ursula Le Guin wrote an essay, I think around 1985, with the title, She Unnames Them. It's just a one-page essay. You can find it online. It's beautiful writing. And the protagonist is Eve in the garden. And Eve goes around the garden and she unnames all the species. The goats are no longer the goats, it's just Bob or Sally. And all the creatures get unnamed. They lose their species names. She takes them away. She removes the species names. So now it's just individuals. And eventually she comes to humanity and tries to have a conversation with Adam. He's too busy and distracted. and basically makes a unilateral decision to give up the human species as a brand. And it's a really interesting anecdote because it raises some questions having to do with how would you feel about modifying Bob? Not Bob the human, but just Bob. And these conversations about, we're presuming a type of species identity and then cultural identity on top of that, and it's not necessarily, that's a construct, right? And it's not necessarily one we have to keep. It returns to how we feel about things in nature as well. So I'll just leave that hanging as a puzzle.